Okay, I think it's 3 minutes past, so even if people are still logging in, we should get started. So it's really great to have you all here when we are ending towards the end of 2020, a year that turned out so differently from what we could possibly imagine when it started. So it's been a year of stress and isolation and also a lot of worries for the unknown for many of us. But it has also been the year when we all, or at least very many of us, learned to use online video platforms. As this is the only way we can really stay in touch. And I have to say that at this time, especially maybe at this time of the year, I really miss you all. And those of you who I know and I've met before, I wish we could be together. But anyway, this is a good compensation. We can communicate and be together in this virtual way. So, and it's also made it possible that we are doing this online to connect with people we haven't met before. So that's also really, really very much appreciated, not just to see you again, the ones I know well and really care about, but also the ones who are in here for the first time. Also, maybe today, I've seen some new names, which is also so valuable, I think, for us and for the community, and I hope definitely for each single one of you. And actually this afternoon, Nira from Israel, from the HD Association in Israel, and myself, we organized a meeting where people from a care home in Norway, the professionals there, could exchange experiences with 10 people from Israel from different care homes there. And I don't think neither Nira nor me would have thought about that opportunity before COVID. So that's also really some value being brought to us all through this crisis. So when we were planning how we could end this weekly series we've had of webinars, actually since September, we wanted to offer you what I was saying, I don't know if that's the right expression, a real steak, so a real good bite. News about HD research is a topic that's always of interest to people and is being requested from you when we ask you. And tonight I'm really, really honored and so happy that we have these excellent presenters. So I have asked Professor Anne Rosser from Cardiff University, who is also the chair of the EHDN, and Dr. Ralf Reilmann, who is the founder and the chair of George Huntington Institute, to tell us what achievements, what progress, what has happened in clinical research in HD in 2020, and what are the prospects, what can we expect for 2021? Because despite COVID-19, so many heroes have worked hard and others stood up by their commitment as participants to continue ongoing trials and to get others prepared and even started this year.
And we also have Alzbeta with us today.
Yes. And we have also asked, this is in my talk, my dear friend, Dr. Alzbeta Mühlbäck to be in the panel to exchange or to answer questions or comments you may have. And she will also give us an input into, I would say, maybe an HD everyday life. And as we are close to Christmas, we wanted to make this a little bit of party. That's why I've decorated with my Christmas decorations behind me. And we have a little other cultural surprise to present to you. Mm-hmm. And it's really amazing because today we are people from all over the world. So maybe I'm missing Australia, but we have Argentina, the United States, Israel, and so many different European countries. It's really amazing, and you are so warmly welcome, all of you. And even my husband, Svein Olaf, from the neighboring room in the house. So this is all from close to really, really thousands of kilometers away. It's amazing. I think we should start. So we will appreciate really to have this an interactive and not very formal format. Those of you who have attended before know that we appreciate if you can comment in the chat, and you can also unmute yourself. And Egypt, I forgot to say, Egypt is also represented. So Africa is definitely represented here. you can post comments or questions in the chat, or we can bring them up also if you unmute yourself. Let's see how many comments and questions you have. But as I say, we really want this to be interactive and being guided by what you think is really not easy to understand or what you are really curious about. So please do that. And then I will hand over the word to, I think, Anne and Ralf together, because you are sharing the presentation. So you manage this between the two of you. So please, the floor is yours.
Thank you, Astri, and good evening, everyone. I will try to share my screen now, and then Anne will start with a little welcome. And I would actually just say briefly that it would be great, and you're welcome to interrupt us when we talk and present. There are lots of issues we'll tackle, and as we go along with this size of group, I think it would just be great to use the digital format to talk directly, which is always a bit difficult in big halls maybe, so we can use this opportunity. Please do feel to ask questions in between. Okay, Anne, do you want to take over?
Yes, thanks, Ralf.
Try to get that. Can you all see the screen?
Yes, we can.
Okay. Great.
Okay, so first of all, thank you very much for asking the two of us to talk. It's a real honor to come and talk to this group. And as Ralf said, we'll do the presentation between us. If we end up talking to each other, please feel free to interrupt and stop that. So obviously it's impossible to talk about 2020 without mentioning COVID-19. It's affected things right across the globe. And as you probably realize, particularly following the early series of lockdowns, a lot of work, a lot of lab work and a lot of clinical work was just stopped in its tracks for a while everybody made things safe and also concentrated on trying to produce vaccines. So this has disrupted clinical studies, but I think it's also really important to say that, it's a testament to the commitment of all, so many PIs in the Huntington field and to all of you, to all the participants, that a lot of these studies are now up and running again. Not all at full capacity, but I think nevertheless, it's a real achievement to go through this and to get the clinical research up and running. Clearly there are vaccines on the way, and we don't quite know how long it'll take for this to really make a difference, but there's some light at the end of the tunnel now. We have the next, thank you. So the last time a lot of as scientists and clinicians saw each other was in person was back in February. And one of the last meetings before the lockdowns was the CHDI Therapeutics Conference in Palm Springs, and we were treated to a whole range of really exciting scientific and clinical presentations. So I think we can be confident that research in HD is healthy and thriving and moving forward. Since then, as Astri mentioned, we've all become quite expert at joining video conferences, and in September, sadly we weren't able to have the face-to-face event, our European Huntington Disease Network plenary event, which we have every 2 years, but we did instead have what we call the bridging event, which was an online event with scientific presentations and clinical presentations, and I think that was very successful. So it's clearly not going to displace face-to-face activities. We do want to hold face-to-face events again, and hopefully we'll do that this time in September next year. But I think we also have learned that actually we can exchange a lot of knowledge in a very useful way on these platforms.
And if I may add to this, Anne, I think it's also interesting that we can reach many, many people, right? I mean, it was always great to have the in-person meetings, but with the digital platforms, we can actually have even a higher audience because people, both researchers, but also patients and their families can just tune in, even if they don't have the time to travel and maybe even the funds to travel, but we can actually reach a lot of people, and you can look at topics and talks and presentations online afterwards, which I think has been used quite a bit as well, as far as I know.
Yeah, no, quite right. So I think, you know, we've got new tools now that can be complementary, even when we can get back to face-to-face meetings.
True.
So I'm pleased to be able to say that Enroll HD, which was also initially badly interrupted by the pandemic, is now largely up and running again, not quite at full capacity. For those of you who don't know about Enroll HD, it's a very large observational study that's been running for quite a lot of years now. 150 active sites in 20 countries, a lot of those countries in Europe, and now over 20,000 active participants worldwide. So in Enroll HD, participants are seen annually for clinical assessments, and there's also the option to give blood samples. And I think it's fair to say this has been a really important study for furthering our understanding of Huntington's, and for clinicians and scientists to be able to get access to these really large data sets to work towards a whole range of things which are necessary for to put in place for really good clinical studies. So this has been a terrific collaboration between clinicians and participants, you know, families. And you to be in Enroll HD, you can be in Enroll HD whether you are gene positive or gene negative, whether you have symptoms of the condition, whether you're asymptomatic, or whether you're just a family member. So it really is a very inclusive study.
I think it's been a major backbone of all the activities in the Huntington field, hasn't it? I mean, I think on the one hand, it's kind of like a rock for families, I believe, because we sense when they come here, they really enjoy their visit once a year because we have time to talk to them, and you touch base with what's going on in research. You get advice on clinical care. So it's really a combination of clinical care and research. But I think it's important for every one of you to understand how important actually I think Enroll HD for us is. For me, it's really the backbone of a lot of the, like the skeleton of a lot of the projects we're doing, because we can actually be much faster. I mean, we all are striving together with you for a better therapy for Huntington's disease. And I think it's been amazing to experience in the last few years what we could actually do. I think 4 or 5 years ago, we had a couple of huge clinical trials running in parallel. We had at some point 1,600 or nearly 2,000 subjects in clinical trials at the same time, I think, and that was unthinkable of 15 years ago. And today, although Huntington is a fairly rare disease, it's still the most frequent neurodegenerative autosomal dominant movement disorder. But anyway, I mean, it's possible to do these things. And that's only possible because you all help. And I think it's extremely important, actually, this Enroll HD project, because it really is facilitating other studies and treatment trials that we'll talk about a little bit in a minute. And it actually encourages sponsors and people to put their efforts into Huntington's disease because they realize it's realistic to do a clinical trial. It's okay to do several trials in parallel, and you can even recruit 800 or 1,000 subjects into one trial. So that's extremely helpful. And whoever of you has the opportunity to motivate people in their families and environment to participate in one of the many centers that we have in Europe or across the world, it would obviously be great if you can be part of that study.
So to just pick up on, I totally agree with that, Ralf, and to just pick up on something you said there, it's important for people to recognize that you can be part of Enroll HD and still be enrolled in interventional studies. In fact, it's actually what we call a platform study. So being part of Enroll HD can be a gateway into other studies if you're interested in being part of them. So the next slide echoes that. So it's a platform study, which also means that we can use Enroll HD to enable other studies. So other studies may use the data from Enroll-HD or use the participant listings or the monitors. They can use a number of different elements of that platform. And a good example of this is the PACE-HD study. So that's an investigator-initiated trial of exercise in Huntington. So there's been quite a lot over the last 10 to 15 years in Huntington's disease showing that increased activity is associated with improved function. So obviously this is a very important message. And what PACE-HD is doing is trying to look at that in larger populations than have been studied before and using a specific exercise intervention. Do you want to just put the next bit up?
Just one question, actually. When I present right now, do you all see Anne Rosser or do you just see my slides? Because I have you as a video here. I don't know if that's transmitted. Do you know what I mean? Or do you just see my slide?
We just see your slide. We don't see your screen. Yeah.
You don't see the videos that I have, like yourself right now. Good. I just wanted to make sure that you can see all the slides.
I can see you.
Okay.
Yeah, but you can't see his video. You see the videos on your own screen and then the shared presentation.
Exactly. Okay, thank you. So I just didn't want to interfere with the presentation because your videos are on top of my presentation here on my screen.
Oh yeah, no, they're separate, Ralph, on the screen.
Okay,
yeah, perfect.
That's the message from the PACE-HD study, I guess, right? Activity is the best medicine. I think we still have to wait for the results of the study, though, and do we? I mean, for
yes. PACE has fully recruited. It's now sort of getting into data analysis, but yeah, it's going to be a little while before we get the results.
I still think we can say with all the centers that have participated that activity is clearly a good medicine for Huntington's disease. We already know that now. To keep active is good. Another study that we wanted to briefly mention that is also added on to Enroll-HD is the HD Clarity study. Many of you might have heard about it. I personally think is one of the most important studies we're currently doing because we're looking at the CSF, the cerebrospinal fluid that we have to win by doing a lumbar puncture. In fact, HD Clarity, I think, has made a huge contribution for many of the sites to be prepared for the antisense oligonucleotide studies, the Roche trial, the WAVE studies that we'll talk about in a minute, because centers have learned to do lumbar punctures well. And why is that so important? I mean, you can imagine that the cerebrospinal fluid is actually around the brain and around the spine. Luckily, it's the same fluid that we can collect in the spine, so we don't have to go with a needle towards the brain, but we can collect a lot of the very good sample in the lumbar area. And you see here on the right-hand side, the different positions, how we can collect the CSF. It's actually not that bad for those people that have already done it. And even some of my famous colleagues have undergone the procedure. So it's in that area of the back, you don't have as many sensitive nerves. So doing the procedure is not really that dramatic, luckily. It is very helpful for us, for the participants that contribute to the study, because the CSF is close to the brain, as I just alluded to, and it gives us very important information about biomarkers, about proteins that we can analyze in this fluid that give us indication about what is going on in the brain, because that fluid is in a certain space that is well protected, and it is more closely connected to the brain than the rest of the body. If we get samples from the blood, it doesn't tell us the same thing than what we can read and understand from the CSF. So for research purposes, the study is great and very important. And we've found a lot of improvements, or we've been able to develop a lot of improvements for assays, such as measuring mutant huntingtin or other markers. You might have heard about NFL and neurofilament light that is being researched as a marker in Huntington's disease as well. Now, this is a way how we actually measure the MHTT concentration in confocal microscopy by using an antibody to mark these green particles, which are mutated huntingtin in this case. And by marking them and putting them into a certain machine, we developed an assay that can actually measure up to a degree of sensitivity in femtomolar areas. So this is very, very few particles. It's important, though, while we can already measure mutated Huntingtin, that we improve these assays further and try to understand what other measures we could use in clinical trials. So HD Clarity, I think, is a very important endeavor. And we've so far had about 400 people that were already participating in the study. There is a baseline lumbar puncture, and then you can volunteer to do another lumbar puncture after 1 month. So you see here in these centers, there were 89 people screened. Here, for instance, 82 people actually took the first test and 27 did the 1 month test. And we have different stages here. We have early manifest, late manifest people, moderately and advanced people. We have controls that can participate as well. And just recently, and that's, I think, is important for the community to know, we've had the pleasure to actually get an amendment of the study accepted, and now we can do annual follow-ups. That's very important too, because we have to understand the dynamics of certain proteins and measurements longitudinally. So please support this study if you can, and go to the centers that do it. Multiple applications for CFS research have been done recently. I'm actually part of the scientific review committee of the Enroll HD study that also grants people access that want to do research with the CSF samples that are collected by this study. And we have multiple, very good examples of how research could be accelerated this way. Because in the past, we had to develop an idea, we had to write a grant proposal, we had to get funding, and we had to get it through ethics, and then find sites and collect samples, and that took maybe years. Now we have a huge biobank of samples, and people that have important questions can come to the committee, ask for access to these samples, and then get research done really quickly. So it's a fast progress that you all make possible. Nevertheless, the samples are not indefinite, so we have to recollect samples, and your support is greatly appreciated here.
Can I comment or interrupt you a little bit? Can you quickly, because I think maybe you have the map, can you quickly tell us which countries this is done? Because it's not for everybody to just volunteer and say you can have my CSF.
It's actually on the HD Clarity website, so I should have probably, there was no map, I have to say, and I should have probably looked that up myself. I can probably look it up later on when we have the Q&A session and give you a bit more information. I know it's done in Germany in multiple sites. It's done in Great Britain in multiple sites. I'm fairly sure it's done in France. I know it's done in the US and in Canada, but I don't know all the sites. There's one in Poland, in Warsaw, I know, and I would have to look it up. But you can look it up at the website that is given up here, hdclarity.net. Just type into Google HD Clarity and it will map up. Yeah, thank you.
Okay, thank you.
Yep. Anne, you want to take this?
Yeah, we'll move on to talking about some disease-modifying strategies. And one that you may well be familiar with is lowering mutant Huntingtin. So the reason for thinking about lowering mutant Huntingtin is that we know that the Huntingtin mutated gene produces mutated Huntingtin protein. And so if you carry the gene, it means that you are producing a certain amount of the mutated protein. You've been doing that all your life, but it has mildly toxic effects on the cells. And I say mildly toxic because if it was very toxic, it would be producing difficulties from very early in life. So the idea of this strategy really is lowering the levels of that protein may give relief to the cells and allow the brain to recover. So if you could move to the next. So yeah, so following on from a lot of animal studies showing that actually lowering the protein levels in animals did lead to functional improvements, a company called Iona produced a method for lowering mutant Huntingtin, which was tested in animals and then moved on to a clinical trial. So this is antisense oligonucleotides.
And these are molecules that we can give into the CSF, and Ralf will say a little bit more about that later on. But basically, we need to get these molecules access to the brain. And then the idea is that they prevent the mutant gene producing the mutant protein. So they result in lowering levels of the Huntington protein. So that's the idea. The Iona study, which ran between about 2015 and 2017, and this paper was published, I think it was last year, beginning of last year. This was quite a small study, showing it was a dosing study looking at over a fairly short time span. So patients were given monthly intrathecal injections of the antisense oligonucleotide, the ASO. So that's through a lumbar puncture in the back. And what this graph on the left shows is that there was a dose-dependent reduction of mutant Huntington protein. So what that means is the higher the dose of the ASO that was given, the lower the mutant Huntington was pushed. So what that trial showed was that the ASO did what it said it would do on the tin. In other words, it did indeed lower the mutant protein in the CSF. And the other thing that came out of this study was that actually that was quite safe. So there were very few adverse events. In fact, almost all the adverse events that were seen in this study were related to sort of more mechanical problems related to the lumbar punctures. So this showed it was safe, showed it engaged with the right target, but it was too small a study to for us to be able to say whether or not this is an effective agent. In other words, does it actually make people better? So Iona sold the license for the ASO to Roche, and this is now called Tominersen. Sorry, I just couldn't get that out. Thanks, Ralf. I'm glad you're here. So this has now recruited to a large phase 3 study. So this is a global study, over 100 centers, over 800 patients. Unlike the Iona study, patients are getting injections either every 2 months or every 4 months, and that may be although you won't know that if you're a patient in this study, or placebo, because this is a placebo-controlled trial. And it's for much longer. The follow-up period is much longer than in the Iona study. So people have been followed up for 2 years. So this study is now pretty much fully recruited. It's running on. It actually survived COVID really quite well. They've lost relatively little data. And I think we, correct me if I'm wrong, Ralf, but I think we're expecting the results of this study probably in early 2023.
Well, let me think. I mean, I think we are about into the first year. So 21, I would say 22, right?
End of 22, maybe?
Yeah, 22 probably.
Yeah.
I'm not wrong. I mean, it's a 2-year study. And, you know, actually, I may add briefly that I think this was probably a world record in terms of recruiting a trial, probably for the whole field of neurodegeneration. So the Huntington community should get a big applause for that. It was incredible how fast the study recruited. Very well organized, of course, from Roche and the Roche team as well. And the whole International Huntington Association supported this very well. I think it's a very important study, obviously, because as you just alluded to, the prior study done by Iona that you just showed the results of clearly showed for the first time that this was a proof.
Ooh. Ralf, can't hear you.
Okay, I was muted centrally.
I'm very sorry, I pushed the wrong button. I didn't mean to. I was going to admit somebody in the waiting room.
You tell me I'm talking too much. Yes. No, I'm just, I think it was really important to see that this proof of concept that was established in this phase 2 study now is being carried on to a really huge phase 3 study that will answer a very important question, the important question whether what we see in biomarkers here that we worked on for a long period of time can actually convey a clinical benefit. And I think that's what really is the important question now. And it's great to see how well the study was recruited and how well it's being maintained and supported. So this is, I think, one of the most important questions the Huntington field has to answer and can answer in the next few years. And I think in a way, we're shortly before Christmas. I mean, aren't we spoiled, I have to say. I mean, looking back at my own career, having worked on Huntington's disease for 29 years now, I'm just thinking, it's amazing that we can answer these questions these days, that we are allowed to answer them, that we can do really, that we can go to the roots, as Anne just said, I mean, or showed in the picture, we can go to the roots of the problem. And we have all these technologies available now. We have the patients, we have the centers, we have everything that we can test these questions. There are many diseases where people would crave for similar trials. And I also have to say that we don't know yet whether this is the final answer to Huntington's disease, but I think it's a very, very good bet that this particular onset may add to change in Huntington's disease and may give us one therapeutic option. It might be important to mention that the approach here is a non-allele specific approach, which means that we actually lower not just the mutated Huntingtin, but also the healthy Huntingtin. What that means, we all don't know yet, but we have seen that if we do not have Huntingtin available very early on in the development of life in animal models, that this is detrimental. So the question will be, how early can we actually use these drugs and how long can we use them and how much can we actually lower Huntingtin? But you know, those are good questions to answer when we see first that there is efficacy. And when we have those problems, they're good problems to have and we can answer them next. But it's important to understand that what we are doing is actually lowering both Huntingtins, the healthy and the genetically modified. And you all remember that we have two alleles. We have a healthy copy and a copy that carries the disease. Okay, just a comment. Anne, please go ahead. You might have other things to say.
No, I think we could go to the next because it runs on quite well from what you've just said, Ralf, I think. Yeah, which is obviously that's tominersen, that's one product. And as Ralf has just said, it lowers both the mutant Huntington and wild type Huntington. So the question is, can we do better than that? Can we do allele specific Huntington lowering? So again, just to be really clear, we've all got two copies of the Huntington gene. If you have a mutated copy, it also means you've got a normal copy. So you're producing both normal Huntington and mutated Huntington. So there are ways of trying to lower just the mutant Huntington. And it turns out that there are little areas on a gene which vary somewhat between person to person. And these are called single nucleotide polymorphisms, or SNPs. You may have heard people talking about SNPs. And there are a number of SNPs which are quite tightly linked to the CAG expansion, which means that you can try and generate an ASO that recognizes those SNPs. And if you're lucky, you may have a SNP that means that you can then sort of tie the ASO just to the mutated gene and prevent only the production of the mutant Huntingtin. So a company called Wave has been working hard on this technology. It's not possible to test this in animals models, or at least it's not possible to test the allele specificity in animal models because animals don't have the same SNPs as we do. But they have been able to test in animals that these ASOs work just like the Roche product. So in animals, they lower Huntington and they produce improvements in movement and in behavior. So if you want to go to the next slide, Ralf.
Somehow a part of my control doesn't move anymore, but anyway, it works through the mouse. Yeah, those are actually examples of the WAVE studies that they have done. And as just Anne alluded to, we don't have animal models because the animals don't carry the same SNPs that humans have. But in cell models, WAVE could show, as you can see here on the left-hand panel, that the RNA for Huntington can be lowered specifically. The blue one here is the mutated Huntington RNA, and the purple one here is the wild type Huntington. So there has been a lowering, as you can see here, which is more dominantly seen in the mutant than in the wild type. There is some lowering also on the wild type, but there is a very large degree of specificity with this particular onset. And that here is on the left-hand side shown for the SNP1, and this is shown for the SNP2 here. Two SNPs that we currently actually try to address in a phase 1 study with WAVE. We have first looked in a study that was conducted in the United States how frequent these SNPs actually occur, because Anne just said that these are SNPs that happen, and some of these SNPs actually are luckily inherited in populations quite consistently because they probably happened long time ago in evolution. But nevertheless, one of the drawbacks of the allele-specific therapy approach that WAVE is pursuing is that it wouldn't be available for everyone straight away. It's a little bit of an individualized medicine, if you wish, already. You have to first test for the SNP. And if you have that SNP on the allele that carries the Huntington mutation, that particular SNP, the SNP1 or SNP2 in these cases, you might be able to treat it this way. However, there's a little pitfall on top of that. If you have the SNP1 on in the place where the mutated Huntington is and on the other allele, you can't do the treatment either, because then you would reduce, again, both. I mean, in a way you could do the treatment, you would end up with the same scenario that Roche is trying, but you wouldn't have the allele specificity, right? You'd lower both. So what you need is on the allele that carries the mutated Huntington, you need one specific SNP that isn't present on the other allele. You have some subjects actually where you have SNP1 and SNP2 on the allele where the mutation is found. So you could use either approach. Overall, we collected blood from more than 200 subjects in US centers, actually Enroll-HD centers. And we did specific analyses, and what we found is that the SNP1 and SNP2, which are the two SNPs that occur most frequently in European populations, is covering for about 60% of the subjects, about 2/3, one can say, can be reached with SNP1 and SNP2 treatments. That's of course important to know, but it doesn't mean that the other patients couldn't be treated with this way, but we would have to use other SNPs and you have to identify them.
So actually,
Sorry.
that's the progress that's been made this year. I'm thinking because we have heard about the WAVE study for quite a while, and it's taken some time to have people recruited in, I understand. But is it possible to say something about status? Where is this now?
Sure. No,
Study,
the WAVE study
in
is
terms of recruitment and how it's really.
The WAVE study is a phase 1 study, so it's a safety study first. We want to assess safety. It takes time to do this, and actually it has a specific reason that it takes maybe even more time than the study that Roche did, because Roche, as we alluded to earlier, could actually use animal models for safety, but we couldn't do that in the WAVE project. I say we because we are a side of the WAVE study as well. I've been involved in the steering committee, the advisory committee of WAVE for about 5 years in the US, and then I eventually had the opportunity to be a site here as well. And UK has sites, and Poland has sites in Europe now as well. The study has expanded to Europe because it's going to be a global approach for treatment also in a phase 3 if this study happens. But we had to start in a very low dose for safety reasons, because the dose, like these approaches hadn't been tested in animal models, because we just alluded to the fact that we don't have animal models that have these SNPs. So for safety reasons, we had to test very, very low dosages first and then go step by step until we could slowly increase the dosages. And there has been a communication recently, I don't have the slide here, but actually in the last dose tested, the last dose before the one that is currently tested, we already saw lowering of mutated Huntingtin in the wave approach with the SNP2. So there were some data shown in recent conferences publicly, so we can share that. There was some lowering already. And now we go next steps of dosages, and this has to be done carefully, of course. You have to wait a certain time until you expose the next subjects. In general, I have to say that I think whatever we discuss here today, there will be multiple approaches, of course, in the future. I personally don't see that as one therapy developing for Huntington's disease, hopefully, but more as a cocktail and probably a multi-drug approach that we can hopefully do in the future and trigger drugs to certain conditions. For instance, this particular approach may be particularly helpful if it conveys clinical benefit in the end to use in early stages of the disease, because allele specificity may be important then, as we just discussed. Whereas in later stages of the disease, maybe allele specificity is not so important. I mean, early I talk now actually pre-manifest prodromal, so very early on. So I think it's important to explore whether the allele specificity has an additional benefit, and it's an important research question for that reason. We do currently Astri 3 studies, actually the precision HD1 and HD2, SNP1 and SNP2. It's listed the other way around here because the precision 2 started first and then the precision 1. It's 2 separate studies for each of the SNPs, and the precision 3 study is in preparation and will probably start in a couple months. So that's the third SNP that will now be tested. We haven't mentioned that yet. There are multiple SNPs that could be tested in the future. As I just said, we cover about 2/3 with SNP1, SNP2, and if we add SNP3, we'll probably cover about 75% of the people, maybe 80. And then we can add more of the SNPs that are known to cover more people. But every person that wants to join this study, that's another drawback, and takes some patience, has to first be genetically tested for the SNP, and that takes some time. It's not a method that you can order in any lab. So it's a very specific message, and you have to be very careful and test that very precisely. And that takes a few months to test, actually. So that's one of the reasons why that takes a little longer as well. We have
Dina, sorry, I have to interrupt you a little bit, Ralf, because you have so much on your mind. Dina, you were saying something, I think I interrupted.
Yeah, I had a question. Sorry, Ralf, can you go back 2 slides?
2 slides, sure. This one here?
Yes.
Yep.
Where is this data from?
This is from cells that have been...
Cells, okay.
This
is not the trial.
No, that's not the trial. And unfortunately, as I said, we don't have transgenic models for SNPs.
Okay.
We cannot go so deep into the details because transgenic it's too complicated in a way. Sorry for saying this. It's really interesting. So there are different SNPs, different people can be included in the study. The precision 2 is finished with recruitment, so that's done in terms of that, or
no, it's still ongoing. Precision 1 and precision 2 are still ongoing. As I just said, we had to be very careful with getting higher on the dosages. So we are at 32 milligram right now in both of these trials. The 16 milligram dose in SNP2 had already shown some lowering of mutated Huntingtin, and now we took the next step, and then possibly there will be other steps in the future to go even higher in the doses. But before you take these steps, you obviously want to look at all the safety and particularly also the efficacy markers, efficacy in terms of biomarker, mutated Huntingtin and others. So that's carefully done. And the study is conducted in Europe, in Australia, and in North America.
But you said there were good results by this SNP drug?
Well, there were good results, preliminary results after the 16 milligram cohort analysis of the SNP2 that was done as scheduled at the end of last year. And it showed already, I think it was 12% lowering. And again, we cannot really compare that so easily to the Roche phase 1 study because Roche could go into the trial with a first dose that was estimated based on the animal effects they saw on the transgenic animals, the lowering. We didn't know how much lowering to expect, and we didn't want to risk to lower too much because that would have been potentially dangerous. So we actually started with a very low dose in the WAVE study, and then we had to escalate, and now we already saw efficacy signals. and now we go up higher, and that's currently done. So the study I would expect will take in, I don't know, it's gonna depending on how high you go with the dose, it will still recruit people. And definitely people interested can still contact the centers and ask to be tested for the SNP if they qualify. Otherwise, of course, the centers have to look at a number of inclusion-exclusion criteria. But I think it's a very important study too, because it would open the door, as I said earlier, to an allele-specific lowering, which may, in my view, be more, possibly easier usable in prodromal pre-manifest stages and maybe in younger people as well, where we have some concern that lowering huntingtin may have side effects, because huntingtin has important functions in the brain as well, in brain development and axonal migration and stuff like that. So it's important probably.
Yeah, I think, Ralf, it's just to pick up on something. I think you mentioned efficacy, but I think we should be clear that we don't have efficacy data.
Efficacy on the biomarker, sorry, not
biomarkers, that's right, not efficacy in clinical terms.
We have no efficacy in any of the ASOs yet. We have to also be appreciative of this. I think it is important to realize that all the efficacy, if we want to call it efficacy, all the changes we have observed, let's say, in all of the studies, that's true for the Roche project, that's true for the WAVE projects, that's true for all the projects that we've done so far, are just biomarker based. We don't have any compound yet that has conveyed a clinical benefit. We have to be mindful about this as well, because I think, honestly, as much as these approaches are extremely important, and they are the hypothesis to be tested right now, we should also be prepared for surprises, because we all hope that this works, but we don't know yet. We have to do the proper testing. In Alzheimer's disease, for instance, the field was absolutely committed to, and very certain, based on preclinical observations in animals, that lowering of beta amyloids would have a clinical benefit. And in the end, that could not be confirmed in huge studies. So we all hope this works, but we have to also be mindful to do our homework and do the studies properly, and that's exactly what we do right now, I think. And as I just said, particularly the Roche trial, of course, it's the very fascinating trial at the moment that will show us probably in a year or two whether we have a clinical benefit that goes along with these procedures that we carefully test these days. But I think it's very encouraging to see that multiple approaches are on the way, and we should probably advance to get to the 2 or 3 other little approaches that we want to show, unless there are urgent further questions. Any further questions at this point?
Yeah, I just have a comment, sorry, Ralf, and I'm going to play devil's advocate here. You would think that after a year of administering the ASO in the Roche trial, that you would hope to see some sort of clinical changes in the participants.
You'll be very careful, because I understand what you want to imply, but I can tell you one thing. As a clinical trialist that I would like to see myself, meanwhile, I have learned one thing: don't trust your own judgment. I think, like in terms of clinician, like as a clinician, some of you know that I've been extremely committed for my whole career to develop quantitative measures of efficacy readouts, and I'm totally convinced that we need objective readouts, both on the clinical side, but also biomarkers, as we just discussed. That's why I'm so much supporting HD Clarity. We were actually one of the top sites up there, because we do lumbar punctures here all the time. Our patients are convinced. We tell them how important this is. We need measures, we need good measures, and maybe NFL, maybe MHTT even is not the measure yet. So we need to have young people come into the field to get their ideas tested and see what alternatives we may have to look at in the future. There's one thing that Anne will allude to in a few minutes, one approach that will be tested. So yes, we don't know yet whether it's the big step, but also we don't know yet whether the delivery is the right one. And that's actually our next slide. So I want to take that now.
I'm a little bit conscious about the time, because we have already over the middle, because usually in these webinars we take a short break, because it's quite intensive for us not doctors also to listen to all these information. So I think it could be good to have a 5-minute break. I don't think I will allow you more because there is still things you want to cover in your presentation, obviously, and you may think of also if you have some questions or how much you want to put in the chat in the break. But I think I will stop you there and take a 5-minute break. Or do you have just one sentence you want to say before we do that?
We can actually restart here. I think that's a good point to restart.
Point.
Yeah.
And sorry
Okay.
it took a little longer, but I was thinking that if we just take the discussion now, why we're going, this is probably very valuable as well, and would really encourage everyone to ask us questions. Any question, please, can be asked, and we'll try to answer it. Okay, thank you.
Okay, so 5 minutes, come back 2 minutes to 8 my time, please.
Okay, can you see my screen again?
Yes.
Okay, so one of the things that we actually just wanted to get to when we talked about the ASOs are a few issues of problems that we may also have with the ASOs. And Dina just said, well, basically she asked the questions, what do we see so far? Of course, the lumbar puncture is an issue that we cannot avoid to think about, at least because delivering lumbar punctures for 20, 30 years is probably a problem. So we want to see if there are other ways possibly to reach targeted therapies as well in a cocktail of available options in the future. Another thing to discuss is actually the cerebrospinal fluid circulation, which you see here. The cerebrospinal fluid is produced in the middle of the brain and then goes through these, we call them ventricles, through small canals as being pushed outside around the spinal cord and around the cortex, and is being absorbed up here. So that's the flow, how CSF is produced, goes through these little canals and goes to the brain and then is absorbed. So we inject these drugs down here in the lumbar space, and these drugs would have to go against the flow of the CSF, which is about 500 milliliters a day. So we take about 10 to 20 milliliters when we do a diagnosis, but we're producing 500 milliliter. So that's a question whether we, using the injection methods we are currently using, whether we really get to the point, to the areas in the brain where we need to go to. So Anne wanted to allude to some.
Yeah, just to very briefly say that, you know, obviously at the moment the ASOs are being given through repeated lumbar punctures, and, you know, there are other therapies that also need to be delivered either into the fluid around the brain or into the brain. So there's a job of work to do, really, to try and work out what are the best ways to do this. You know, if we're lucky enough to have one of these trials become positive, what's the next step after that? Are we going to keep giving people lumbar punctures over years, or are we going to find devices that will allow us to give the treatments more easily? So there's quite a bit of work going into that at the moment. So this is through the EHDN Advanced Therapy Working Group. So it's just bringing experts together, really working on gene therapies and ASOs and cell therapies to try and use everybody's knowledge to try and get to a place where we have better devices for developing these treatments.
And this is actually an example of this. So as many of you know, we have just started the first gene therapy trial in Huntington's disease with Unicure called GIN and TREX1. What we do basically is we want to do the same thing that we do in the Roche and ASO trial and the WAVE trial. We want to lower huntingtin, but we don't do it by repeated injections through the lumbar area, but we actually deliver a virus that is basically used as a packaging system. And inside the virus is viral. It can go into the neurons. It can spread. It's, so to speak, the door opener for us into the brain. The virus is delivered directly into the brain by neurosurgical operation. And the virus carries, like a package, a DNA information. And the DNA then is integrated into the cells in the body, in the brain, and it produces the agent that then lowers huntingtin. It's basically a similar approach than what we do with antisense oligonucleotides, because they kind of go to the construction plan, the RNA, and they shred it. They reduce the number of these copies and then reduce mutated huntingtin. So we do the same thing here, but we do it with this virus. The virus load can be determined. This virus doesn't replicate as other viruses do. So the COVID-19 unfortunately replicates, this virus doesn't. We can exactly say how much we inject into the brain, and we go to the putamen and caudate nucleus to do this. And how this looks like is shown here. This is the area where we actually want to go, the putamen and the caudate nucleus. And we use MRI to plan in every patient individually the access to this area of the brain very carefully to avoid vessels, arteries and bleeding. And you see that then stereotactically an injection is placed here in the top of the caudate nucleus and the putamen. And there's another injection here and then another injection here of a certain volume that we adjust to the size of these structures in the patients. And then these viruses spread in a certain area and they transfect these areas particularly. If they spread further, we don't know yet. We have an example here in a non-human primate rhesus monkey where you see how this is injected with gadolinium and then it transfects certain area. So that can be done. We've done multiple preclinical testing and we've shown, particularly with the mini pigs that many of you know I love very much, we've been able to show that when we inject that in mini pig brain, it actually transfects the caudate nucleus area and the putamen, and it also lowers Huntington. I'm not going to go into the details here, but this is the similar approach that we showed with the Roche trial. So there is a first phase 1 trial ongoing for safety in the United States right now, where about a total of 26 patients will be randomized to get a low dose, a higher dose, or a sham surgery. And if this is all safe, these people will later on get the opportunity to also get operated in open label studies. We'll look at safety first here, at duration of the vector, how long the virus persists and the effect. And we also look at clinical endpoints, exploratory, at biomarkers like MHTT, MRI, but also at clinical scales and quantitative measures like Q-motor. This is a 5-year project, so it's a very long project, but it's really the first gene therapy trial that has come to Huntington's disease, and potentially, as discussed earlier, would give us an approach to a one-shot therapy. If somebody wants to read more about it, the Huntington Study Group in its pamphlet HD Insights published a story about that recently. The whole journal was filled about this topic, and I think it's an exciting alternative for us to see if protein lowering is really efficacious, because this clearly, I think, pretty much shows or will be a proof of concept for actually reaching those brain areas where we want to lower Huntington, certainly, and caudate and putamen, and then we can see whether that gets a clinical benefit to us. And maybe in the future, we can combine ASOs with these approaches to reach enough areas of the brain. So Anne, briefly for you.
I think we can skip this one.
Yeah. Okay.
So I don't know how much time we've got, Astri.
You can continue for a little bit longer, 5 minutes.
Okay, so this is just another disease-modifying strategy, a completely different strategy to the Huntington lowering. So this is looking at tackling somatic expansion. So first of all, we know that if you to develop Huntington's disease, you have to have the CAG expansion. And I think probably many of you also know that the longer the expansion, the earlier the condition starts. But and you can see that on this graph here. But in fact, this is very much skewed by the very long repeats. And if you look round about, I don't know if this is still animated, Ralf, or not. If you, maybe not.
Not
no, don't worry about it. If you, no, it's not. If you just go back, if you look around about 40 repeats, for example, each of these green dots is an individual, and you can read off on the left-hand axis when they started the disease. So you can see that people with 40 repeats, within this population, you've got people starting under the age of 20, and you've got people starting in their 80s. So it's a huge range of different onsets, and not only onsets, but they're for an individual. Well, actually, I'm going to leave this next bit, Ralf, in fact, and go on to the next slide. And really, what mechanism underlies this massive difference in age of onset for a single CAG repeat expansion? And what seems to underlie this is that although if you measure CAG expansions in the blood, they're pretty stable. So if you measure CAG expansion in your 20s in the blood, it will be pretty much the same when you're in your 50s or 60s or 70s. But that isn't so for all tissues. So if you measure CAG expansions in the brain, they're actually getting longer over time in someone who has the mutant allele. And when I say longer, that can be hundreds or even thousands of CAG repeats. And if you look on the right here, you can see a patient who's about 13 years before predicted onset. So this is someone who's asymptomatic. And what this gel is showing effectively is that there are already in the brain CAG expansions I'm talking about in the DNA. So this may, these massive expansions are occurring in areas that matter in Huntington's disease. They're occurring in the areas of the brain that are affected in Huntington's. So we think they may be quite important for pathogenesis. So what's underlying that? If you go to the next slide, Ralph, there's been quite a lot of work trying to understand what's actually causing this massive expansion of CAG repeats in the brain. And this is work that was actually from the Enroll platform and the registry platform before that. So this is a genome-wide association study where the investigators looked across thousands of people with HD and tried to find areas that were associated with great variability in age of onset. And they found a number of areas in these studies that are associated with variability in age of onset, and they seem to be enriched in DNA repair genes. So you might say, well, what on earth has DNA repair got to do with CAG repeats that are getting longer and longer and longer? So if you go to the next slide, Ralph, a possible mechanism underlying this is that when you have the Huntington gene with a CAG repeat expansion, it forms some odd structures in the cell. So you get these hairpin bends and some other slightly strange structures. And the DNA repair system is a system that you have in all your cells to try and keep your DNA in good shape, because obviously your DNA is really important for how your cells function. And it sees these hairpin bends and it thinks that there's a problem. It's something there that it needs to fix, and it tries to fix it. And in fixing it, it makes it longer and longer and longer. So it appears that the DNA repair genes that are very important in our bodies may be a mechanism underlying these long expansions. There's some evidence that for some of these genes, lower levels of the gene can be associated with higher levels, higher age at onset. So obviously this has led people to thinking about, well, can you then knock those genes down? Can you actually use technology such as the antisense oligonucleotides that we talked about earlier to reduce the levels of those DNA repair genes? And would that lead to a reduction in these massive expansions? And maybe that's one way in which we can slow down onset of Huntington's disease. And so this is a strategy that Triplet Therapeutics is taking. This is at the moment very much at the preclinical level, but to my mind, it's a very exciting potential way forward, and it's something that you may hear more about over 2021. And we may, if this turns out to be as successful as we hope it will, it may go to clinical trial, possibly towards the end of 2021, but I think probably more likely 2022.
Yeah, I think this is a fantastic approach, actually. And I think it gives us a therapeutic alternative if protein lowering shouldn't have that kind of clinical efficacy as we currently hope it will have, or if we have problems to actually deliver it. Although here we have to use ASOs potentially as well, so we'll have a delivery issue also. But I think we'll learn as we go along to improve that as well, as Anne alluded to, so maybe other methodologies to deliver. I think we should probably come to an end. I just want to briefly mention as a last point, possibly, that actually together with Anne and with Andy Fagan and Sandra Kostic, we are doing a clinical trial now, so this is not just talking about it, but we will test again the compound predopidin earlier called ACR16 in a study called Proof HD. We want to prove actually if predopidin has a positive outcome on function in Huntington's disease. We've done 2 studies in the past where we've seen that there were motor benefits on the total motor score, 2 big studies, but these 2 studies didn't use the total motor score as an endpoint. It was a bit paradoxic for us as clinical trialists. We had significant changes in this total motor score, but a modified motor score that lacks a few portions of the total motor score was used as the primary endpoint. So formally, these were negative trials. The efficacy was seen in 2 times 45 milligram at that point of predopidin, and we then did a predopidin dose escalation study, PRIDE HD, and in that study, we did not see a change between the treated people and the placebo group on the total motor score, but we saw a benefit in function. So the people treated with 45 BID had this profile on the total functional scale, which is used and enrolled in other studies, and the placebo group got worse. There was benefit also here on the motor measures that were taken objectively, the Q motor. So the placebo group got worse here, and particularly the 45 milligram group maintained function. So there's a possible explanation for the TFC effect here. And we now do a huge clinical trial. Luckily, I think we can now answer, hopefully, finally, the question whether predopidin conveys a benefit in Huntington's disease. It now just starts, and it compares 1-to-1 now, 45 BID predopidin against placebo. And it actually takes into account the COVID-19 crisis already, because we have a couple of virtual visits, and we really encourage people to help us recruit this trial. 440 people will be treated for about 1.5 years, and many of the European countries will participate, and also North America. Just one thing, we'll have oral drugs. Novartis has an oral drug that probably goes into the clinic next year, trying to lower huntingtin as well, not going in the data now, and PTC therapeutics as well. So there are 2 approaches here that use similar mechanisms that modulate the genes in a way that also the mutated huntingtin is degraded and protein is not produced. And these 2 are oral approaches. If they work, of course, we would have a delivery problem less, as we just discussed. And I don't know, Anne, if we want to skip this now here.
I think we do. I'm sorry, we do, because of the time.
Thank you very much for your attention, but I have to say one word still. I have to say thank you to everyone in the Huntington community, and many of you on the phone helped. It's been a hard year for everyone with COVID-19, but actually for us, it's been a dream year as well, because we opened the George Huntington Institute in its own building this year, and our patients and families are so happy to have a harbor to come to for the next, hopefully, decades, and maybe longer, hopefully. And you know what? This was only possible because of the great friendship we have with all the families, with all of you in the Huntington community. And Astri, you have visited, and Swain, Olaf, and you were here. We started with a very, very small 4-room, lightless rooms, actually, department in the university. And we have a huge space now to offer to patients. This only worked because we all work together. So please, please support all your local centers so they can have similar institutions in the future to take care of you and your families and bring advance to Huntington's disease. I have seen so many things blossoming over the last 10, 20 years in Huntington's disease that I'm so certain that we'll find a therapy. We might be very close, but even if we don't have the hit yet, I think we're getting very close. Help us all, and we should get there. So I'm finished.
Thank you. Yeah, thank you, Ralf. We are not closing the webinar yet, but thank you so much. I would like to try to summarize. So the Roche study will be ongoing next year. We will probably not hear anything because it's really in the duration of the 2 year. The WAVE study, these early phases, we might hear from them. I think that's what they promised us, that they will share some information like we had from Roche late in the year of 2017, which was really exciting at the time. So Unicure will continue to recruit, probably only in the United States, or
that's currently the plan. Although I have to say, we've had hopes to get them to. We will try to convince them to do a study in Europe as well, because we want access to patients to this therapy in Europe as well. But
so maybe, but anyway, Unicure will continue the study with the early phase and the few cases, but potentially could come also for some Europeans to take part. Novartis, what are you saying about them for next year? And do you think will they start?
Probably next year. Yeah, I think that's their plan. Fingers crossed.
Yeah, fingers crossed.
Yeah.
And also PTC, because those are simpler in the sense that they are providing the medication via the oral, so taking a pill basically. Yeah. Is that really covers the most? I mean, from the medical.
I think that's a very good summary, Astri. Yeah.
Okay.
And the triplet trial may come.
I think the following year. Probably.
Maybe the following year, yeah.
Triplet, and you also mentioned Proof, obviously, Ralf, who is really being
Proof is
would
already
start early next year already, I think. Yes.
It's already recruit in some countries. Yeah, it's already starting.
Yeah, great, really exciting, and it really demands a great effort from all of us to succeed in doing these trials and learning from them. So I really want to pass on the word to Dr. Alzbeta Mühlbäck. We didn't really manage to organize a panel as we had originally thought, but this was a good way to do it also, to have some interaction from you and input. And Alzbeta, you can please unmute yourself. And I know you have a presentation. I think you have a short presentation. I want to ask you, Alzbeta, because
Yes.
Tess from Sweden and I'm from Norway, and there are many people here from countries, Slovakia, Egypt,
Yes, yes.
countries where no trials are being done actually, or offered to
I
us,
know.
because we
It's
see
very
that as
difficult.
advantageous.
I know.
So can you comment on that? Will it be like that forever?
You know, I hope it's not going to be like that forever. We saw this similar situation in Czech Republic. It's luckily it's changing. There are some trials. Prelenia with Proof HD is also coming to Czech Republic. So I believe we need to keep going to take care of our patients, to build the infrastructure. And as soon as we have infrastructure, and I'm sure that the trials are coming. And if the trial is not coming, we just need to have a finger crossed for the other countries, because once the medication is proved, it's coming to all European countries. So I think it's like a marathon run that we are running all together. So if you are not able to be on your own, be running, then you need to cheer for the others and to get them to get to the, let's say, to hit the button. And I'm sure that we will have a benefit of all of us of it.
Thank you. That's
Yeah.
really cool. Yeah. We want to take part, all of us, or many of us, but
yes,
still,
I know.
we have good people in the race, which is
yes.
the most important. Yes.
You know, I just had a webinar just a while ago with the Slovak Association. It was our first webinar this year, and we were talking about the new approaches in HD. And for sure, those questions are as well coming, and people are asking why we are not the ones participating on the trial. But it was in the same way we tried to explain that you are not the one on the trial, but for sure you are one who is getting the therapy. So now we need to think in the way that other people are taking part in the trial for you. And we are like doing it all together. So you need to keep yourself like very healthy, keep continue exercise, do the things that you can do. And there is some kind of things that you can do on your own to keep you healthy, physically and mentally. So you are ready for the first medication or the first trial that is coming to your country.
Thank you.
And
very
also,
good.
You know, also my presentation today, it's a little bit different because I was supposed to talk. Oh,
sorry, I need to make you co-host before you can share.
Oh, okay.
It's nice. It's really nice to have this kind of power. I can mute people and I can use it.
You can do everything today.
So now I hope you're able to share.
Okay, so I think now I am able to share. Yes, now it's working. So I get this one. Are you seeing the presentation at the moment?
Yes.
Yes.
Yes. Okay. So today I would like to talk about the new therapeutic strategies to face HD in everyday life. It's not going to be a long presentation. I have like 6 slides, and those slides are dedicated to patients that we are seeing every day. And these patients are giving us also some kind of power and the meaning of the work, and they are facing with a smile HD and doing it like every day, and they are never losing hope. So please. So those are all the patients we were seeing in December in our clinic. They are all trying to do the best, because I told them we are having a webinar, so you need to put the best smile ever. And this is the intervention that can be done in each clinic. So those are the best smiles of our patients. If we continue, these pictures have been made by this young lady today in the morning, because I told her I would like to put her on the webinar because she's facing juvenile HD. And she wrote me, don't put the pictures from the clinic, I have the better ones. So she did photo shooting to be today here with you and with her small sister that is giving her a lot of energy, and also the 2 dogs. So, and she's all the time telling me, you know, life is beautiful. And I think this kind of energy we also need to think of, that the people are having and like just sharing with us. Then I have these 3 young ladies. They are like, I'm sorry that I'm not having a music, but those ladies were like dancing all the time in our clinic. You know, they were like the one in this one. She is a dance teacher. She had to have a dance private school. So she was all the time telling me the best physiotherapy is samba. So, and they are all the time telling me, we love music and you just need to dance, and this is only like the attitude. So please share this attitude of these best ladies ever from Bavaria about the dancing samba and enjoying the life. And also this young athlete that is also had a very difficult life. Unfortunately, I put him to this presentation because he passed away in December, but he was fighting to the last minute and was working very hard, and he was really a motivation to others. And I promised him that I will present him once as the best athlete of the world because he was running like till his last minute, and it was always, the game was always on. So always think of this, that there is always a way, and never give up and just fight until the end. So this is from Andreas. And this is my dreamy boy that is always dreaming of something, and he's dreaming so nicely all the time. And he's very young, 18, but it's just, you know, it's just perfect to also have this kind of young patients that are seeing of severity of HD, they are seeing the world in completely different eyes and giving the energy to other ones in the department and in the clinic, and saying always, I am cool, everything is very cool, and you are cool, just relax. And this is something that we need to hear at the end of the year, just to relax and to stay cool.
And I'm sure you love me. I like that one. We should assume that much more.
Yes. he basically thinks that all young girls love him.
Oh, I'm sure he's right.
But you know, if you look these eyes, you have to love him, you know?
Yeah.
You cannot say no.
I'm not a girl, but anyway, I think he looks nice.
Yeah.
So he's really great. And this was a special picture for you all. Because we are supporting other rare neurological diseases, I couldn't discriminate those people that are visiting our department with neuroacanthosis or dysphagia. So please also, I think we need to be as a community to hold together, and the other rare diseases have even much difficult life. Like this kind of neuroacanthosis has a new mutation with a prevalence of 1 of million. So it's quite difficult life. So there is not many therapeutic approaches coming to him, not many trials, but he stick to the HD community, and he's perfectly fine with us. And so
he's welcome. You can tell him.
Welcome.
He's welcome for sure.
So he's all the time telling me it's so unfair that HD is having so many trials and we don't have any. So this is the different part of the life. So those are our guys. And you need always to have a perfect stuff. The Christine is just about to go home. So she was telling me the best approach is if you don't see the problems, the problem are not seeing you. So I don't see you, you don't see me. That's the best way if something is not working. Some of those are saying, oh my God, this is not working. So this is our, let's say, our crazy team and crazy stuff, but we need to, in these very difficult times with Corona, what is making our life more difficult, we need to keep some kind of good, good mood and to stay on the track. This guy, he was not really, he was telling that we cannot do the rabbits in December. He said that it's not the Easter, so he is not participating. And the last picture that I have, we had the secret agent in the clinic. Do you know him? For a visit? So for those ones that got the name of our guest, there maybe should be a prize from EHA or something. So as you see, there is always a way, and we need to just enjoy it. And I would just say we just keep smiling for this great year with the European Huntington Association for very nice webinars that really contribute to the HD community with a lot of interesting topics. And thank you that I was allowed to be a part of it.
Thank you so much, Alzbeta. Thank you for sharing those faces and smiles, and humor is definitely something I also really think is helpful for us to make use of. So before you are leaving, all of you, I know some of you have to leave already. I want to share a video because many of you, not all, but many of you were in Bucharest last year, and we had Livy from Germany, from a Huntington family in Germany, a musician making a song for us. Do you remember it? It's called North Star. So I will hopefully share my screen with my Facebook. There it is. Share computer sound.
You want us to sing along?
You can sing along. You can unmute yourself and sing along with the voice you have. So let me see if I can. Can you see this now?
Yes.
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 Hello, everybody. I hope you're all well. Astri invited me to join this webinar tonight, but unfortunately on Wednesdays I have to work late. So I pre-recorded this message and song. Some of you maybe remember North Star from Bucharest, so please feel free to sing along with me. North Star shining so bright, twinkling in the night. North Star glowing so gently, guiding us from afar. We keep on looking back. celebrate Christmas. I wish you all a beautiful Christmas time and all the best for next year. And please continue to do your great work for the HD community and spread your light. And yeah, hope to see you sometime. Bye bye, all the best.
Is that on YouTube, Astri? Is this on YouTube or somewhere?
No, she sent it to me, but it will be now in the recording, so that will be available. And Anne, since you are chair of the executive committee and AHDN, I hope that we can sing this song at the big conference in Bologna, hopefully when we will meet there in
yeah, we should.
Wouldn't that be nice?
It would be wonderful. Yeah.
Yes.
And don't forget, Anne showed us the data about exercise. There needs to be some dancing too, right?
Yeah. And we should be, you know, samba or salsa or whatever. That's really nice. So we are over time, and I wish you all really a nice celebration, whatever way you can, capacity you can, and enjoy the holidays. And we will certainly keep you updated in the year to come, and we hope to see you all. And we will also make some new webinars, but not every week. So we'll keep you informed on what will happen.
Thank you.
I will also give a small applause to you, Astri, for hosting all these webinars and doing a very good job.
Thank you, Mike, and to you for being here and contributing. It's amazing.
Yeah.
Thank you so much.
Thank you, all of you.
Lovely to see you.
Happy holidays, everyone. Bye.
Yes.
Bye.
Bye.
Bye. Bye.